students, this is Dr. Sujata Magdum, Assistant Professor in Zoology, KTHM College, Nashik. Today, I'll be dealing with Immunology for MSc Part 2 students for the semester 3rd, and in this, I'll be talking on Chapter 8, that is Immunotechniques. In this, today we'll be understanding the antigen antibody reaction. With the previous concept, what you have understood in earlier lectures, you know that antigens, they are the foreign particles which are responsible for cause of the disease, whereas antibodies, these are generated when the antigen stimulates the B cells, and these B cells, they get transformed into the memory cells or the effector cells. The effector cells are nothing but the plasma cells which are responsible for synthesis of antibody. So whenever an antigen enters into the body, that time there is an interaction between the antigens, which is the foreign material, and the antibodies, which is synthesized by the host cell. As a result of this interaction, there are large number of reactions which are seen generated into the body. This happens because the antigens, they are very specific, and this specificity leads to a specific stimulation with reference to the antibodies. This is only because the antigens, they show presence of epitopes onto their surfaces, whereas the binding site of the antibody is the paratope. In other words, you can remember that the interaction of antigen and antibody is via epitope and paratope. Now, this interaction is the basis for the development of humoral type of immunity, or it is also called as antibody mediated immunity. This interaction between antigen and antibody is very essential because it helps in detection of diseases, and such in vitro reactions are defined as the serological reactions. These serological reactions enable us to define the presence of disease or the kind of infection which is seen present into the patient. Now, this antigen antibody reaction is seen occurring at three different stages. That is, the first one is with reference to formation of antigen antibody complex. As a result of formation of this antigen antibody complex, there are certain visible events which are seen occurring, like precipitation, agglutination, etc. And as a result of this precipitation or agglutination, which happens to bring together the presence of antigen and antibody in close vicinity, leads to destruction of antigen or its product, hence causing the neutralization reaction. So, these antigen antibody reactions, they have four different salient features, namely specificity, formation of immune complex, binding site of antigen, and binding force of antigens. Let us see them in detail. To begin with, the specificity. Specificity refers to, or the term is derived from specific. Now, what specific nature I am going to talk about is the binding site of antigen and antibody. Please concentrate here. The Y-shaped structure is defining the structure of antibody. Now, this antibody, it has two free ends, and these two free ends are called as the antigen binding sites, or it is called as the CDR regions. Now, these CDR regions are responsible to bind with the antigen, and as I said in the previous slide, that the binding site of antigen is called as epitope and the binding site of antibody is called as paratope. So obviously in these regions there is presence of paratope which will be seen binding with the epitope of antigen, and this leads to formation of a specific type of reaction. Each antibody binds to a specific antigen. This concept is similar to the lock and key arrangement which you have studied with reference to enzyme. There is a very specific key for a specific log. In the same manner, a very specific antibody is seen present for a specific antigen, and this specificity leads to development of the next structure, which is called next point, which is called as immune complex. Now here you can see I have shown three different types of paratopes, and these are the epitopes which are seen present associated with the structure of antigen. Now because of their specificity, they can bind, or you can say epitope can bind with paratope. Like here there is the graphical representation of epitope, which is triangular and is seen present into antigen, and it will be binding with the paratope, which is inverted V-shaped structure. Likewise into the second, here you can see the epitope which is oval in shape. Obviously this epitope should be seen in association with antigen, and this epitope, it will be seen binding with the paratope, which is inverted U-shaped structure into the antibody. So where are these paratopes located? They are seen located at the free end of antibodies, correct? Now here you can see there are different types of antibodies, and these antibodies, they have different epitopes. The first one is the structure for IgD, E and IgE. These structures you are going to study in detail in the next lectures. Now here what you can see, the free end is the paratope. So these paratopes will be binding with specific type of antigen, right? Now this is the cartoon representation of specificity shown between the antigen and antibody. Here these are the antibodies which are seen present onto the specific cells of the body, and these antibodies, they will be binding with the epitope. Now green color is an antigen showing an epitope which is having a crown shaped structure, and this will be seen binding with the Y-shaped structure of antibody. Now here the binding is not prominent because the structural specificity is not observed, but here you can see because the epitope and the paratope, they are similar to one another, hence this antibody, it can bind with the antigen. In the same manner, here there is no specificity seen between the epitope of the antigen and the paratope of the antibody. So binding is not observed. Is that clear? I hope this will help you to understand the specific nature of binding of paratope with that of antigen, that is epitope. Now let us talk about the immune complex. In the previous slide, I said there is specific binding of antigen to antibody. Now this specific binding has been resulted into formation of a large complex, and this complex is called as the antigen antibody complex. Now this antigen antibody complex, or is also called as the immune complex, it is formed because of cross-linking. Here you can see that not a single antigen is seen binding with a single antibody, but a single antibody, it can bind with large number of antigen. That means it is bivalent in nature. Why do I say bivalent? Because here the single antigen, it has the potential to bind with two antibodies consequently. When such is the condition, that means when it is bivalent or multivalent. So what is bivalent or multivalent? If the antigen is bivalent or multivalent, it results into formation of immune complex, and this is formed due to integral binding of the antibody to the soluble antigens, and this has accomplished into formation of antigen antibody complex. So what you should remember, for formation of immune complex, there are two important structures which are needed. The first one is the antigen, and this antigen should be multivalent or bivalent in nature, and it should be in soluble condition. When these soluble antigens which are multivalent in nature come across the specific antibody, there is the binding seen between the paratope of the antibody to that of epitope of the antigen, which is, you can say, mustard yellow in color, and this results into formation of immune complex, and this immune complex is the second step involved into formation of antigen antibody complexes. Now this is the pictorial representation, cartoon representation of antigen antibody complex. When there is no association of antibodies with the antigen is very happily residing into the body. But as soon as the antibodies they come in contact with the antigen, it gets to know its lifespan or its outcome, and so it becomes afraid, you can say. This is just a lighter note of the reaction, I mean to say, right? Now the third one is the binding sites of antigens we will try to understand. Now here in the previous slide I was talking about the epitopes and the paratopes. So what are epitopes? Epitopes are the binding sites of antigen. Antigens, these are foreign material. It could be bacteria, it could be viruses, it could be any parasitic worm or so on. So these foreign material onto their surfaces, because they are proteinaceous in nature, they show certain receptor molecule. So these receptor molecules which have the affinity to bind with the antibody, they are termed as epitope. So in short, you can say that antigens, they show the surface binding receptors with antibody and they are called as the epitope. Likewise, if the binding site of antibody I have to describe, then the free end of antibodies, these antibodies are once again protein molecules. So this free end, which is the amine terminal of the antibody, they show the binding site, and these binding site, they have a very specific shape which coincides with the shape of the epitope. Thus, these binding sites of antibodies, they are called as antigen binding site. Why are they called antigen binding site? Because they have the affinity to bind with the epitope of the antigen. Therefore, they are called as antigen binding site. So, what is the other name for this antigen binding site which is associated with antibody? They are called as paratope. So, now you should be very clear with the two terms, that is epitope and paratope. Epitopes are the binding sites of the antigen, whereas paratopes, they are the binding sites of antibodies. These paratopes, they are also called as antigen binding site. And where are they located? They are located at the free amine terminus of the antibody. This structure of antibody you are going to understand on the LMS lecture which I will be uploading shortly, right? The interesting part of epitope is it can recognize it can be recognized. These are recognized not only by the antibodies, but they are also recognized by the receptors which are seen onto the surface of B cells and the T cells. Now the fourth point is binding force of antigen. So once the antigens and antibody, they come in close vicinity, there is a close association which leads to binding, and this binding has resulted into formation of antigen antibody complex. So the question arises, what makes this antigen antibody remain into the same state? It is nothing but the binding force of the antigen which enables the complex to remain stable for a longer time. And for understanding this, we can say that this binding force of antigen antibody, it depends on three different parameters. That is, the close association between the antigen and antibody, it plays a vital role. More closer is the antigen and antibody, the strength or the bonding will be much more larger as compared to the association of antigen antibody which are seen at far off distance. In other words, you can say the close proximity of antigen and antibody leads to bringing about more association between antigen and antibody. Apart from that, when these antigen antibody they are seen closer together, there are large number of non-covalent bonds or intermolecular forces like the hydrophobic bonds or the hydrogen bonds or the ionic bonds or electrostatic attractions, van der Waals forces, which keeps this antigen antibody together for a longer time. And lastly, which is very important, we can say the affinity of the antibody. What does it mean? Affinity of the antibody refers to, as I said, there are specificities seen with reference to binding of a specific type of antigen to antibody. Obviously, in other terms, you can say the closeness is totally dependent on the structure of epitope and the structure of paratope. Or if the structure is very similar, we can say the antibody has an affinity to bind with the antigen, right? Now, as I have said that these antigen antibody reactions, they are reversible, specific, non-covalent biochemical reactions, and they totally depends on hydrogen bond, electrostatic bond, van der Waals forces or hydrophobic bonds. Now let us see what are hydrogen bonds. These are the chemical bonds in which a hydrogen atom of one molecule is attracted to an electronegative atom of another molecule. All these things we have already studied in biochemistry. Then electrostatic bonds, these are the valence bonds in which two atoms, they are attracted by electrostatic forces, and this leads to transfer of one or more electron between the atoms. Van der Waals forces, are the forces which are seen acting between the non-bonded atoms or the molecules. And the hydrophobic bonds, these are the attractive forces between molecules due to the close positioning of non-hydrophobic portions of the two molecules. And they can be represented by the formula as AB referring to antigen antibody, and Ag referring to antigen. So antigen plus antibody, it will lead to formation of antigen antibody complex. The forward reaction, that is antigen when it binds with antibody, it has K1 as the constant of association. Whereas the backward reaction, wherein if these hydrogen bonds, electrostatic bonds, van der Waals forces or hydrophobic interactions are weakened, that will lead to splitting of antigen antibody complex, and this is much more dangerous because the purpose will not get solved. What is the main purpose of having antigen antibody interaction? This interaction enables us to identify the type of antigens which are seen present into the patient, or it is a serological test. So unless and until the serological tests are performed, one cannot come to the conclusion of the type of disease the person is suffering. So we can say that this dissociation is sometimes seen, which gives us the false results, and this dissociation is represented by constant of dissociation, that is K2. Now let us see what is affinity and valence and avidity. These are the three factors which are very prominently used when you are talking about antigen antibody reaction. So what is affinity? Affinity is actually the strength of the reaction between a single antigenic determinant and a single combining site on the antibody. What does it mean? It means that suppose if I have in the previous slide, as I have shown you, the antigens with a green coloration and three epitopes, they are seen associated. So out of those three epitopes, the binding ability of one single epitope to the binding ability of the one paratope of the antibody is called as an affinity. I hope it is clear to you now. And then this can be also said to be an association constant for binding and is represented by Ka. So what is Ka? Ka is equal to the constant of association, that is K1 upon the constant of dissociation, that is K2. So K2 should be always less than K1. Then here is the pictorial representation. So what you can see here, this is an antigen whose one single binding site, if I consider this lower one, its binding with the antibody will give me the term that is called as affinity. If it, and how is it obtained? It is only because of the structural similarity between the epitope of the antigen with the paratope of the antibody. Hence, if I say that all the epitopes of my antigen, they are seen in close binding with the antibody, we can say there is high affinity between the antigen and antibody. Opposite to that, if the association between the antigen and antibody is not strong, because it could be structural indifference between the epitope of antigen and paratope of antibody, then in that situation we can say the antigen antibody affinity is very low. Now this antigen antibody affinity, it depends on the number of epitope, which is called as the valence. So to have the high affinity, the valence should be greater. Now what is avidity? Avidity is the collectivity or collective term between the affinity of multiple binding site. Means if I consider only one single binding site of antigen and antibody, I can call it as an affinity. But if I consider all the binding site of antigen interacting with all the binding site of antibody, then the term is avidity, right? For example, here if I consider only the lower one, it will give me affinity. But if I consider all the 5 binding site of antigen to 5 binding site of antibody, I can say it is avidity. So here also same holds true. Is that clear? Now let us see what are the different types of antigen antibody reactions. They are precipitation, agglutination, then neutralization, opsonization, then antibody dependent cell mediated cytotoxicity, which is also called as ACDD. Then the fifth one is the complement activation membrane attack complex. Now this is the pictorial representation of antigen antibody reaction. Just now as I have said that it could be neutralization, agglutination, precipitation or complement fixation. So what is neutralization? From the word itself neutralization we can make out that whenever an antigen binds with the antibody, this interaction leads to bringing about the action on to the antigen. So what is antigen here? I have considered the antigen as a virus or the bacterium. So when the virus or the bacteria is nullified, its action is nullified, the reaction will be defined as neutralization. This is seen when there is association of antigen with antibody. In the same way, the next one is agglutination. So in case of agglutination, what you have to remember, large number of bacterium or any antigen, when they are seen in close association with the antibody, it leads to formation of a clump. So this is called as formation of agglutin or formation of reaction, which is called as agglutination. At the same time, if the reaction is between the soluble antigen and the antibody, so as a result of which the soluble antigens, they will be made to settle down. That means they are precipitated and the reaction is called as precipitation. So whenever there is neutralization, agglutination and precipitation, this will enhance the activity which is called as phagocytosis, and as a result of which the antigen will be nullified or it can be removed or it could be brought to lytic action, and this is done by means of large number of macrophages, dendritic cells, natural killer cells and so on. And in case of complement fixation, there is an activation of complement, and this activated complement, it leads to bring about the action of infected cell, and that is cell lysis. And as a result of this, you can say once again the antigens, they are been knocked down or they are removed from the body. Now let us see in detail the reaction, that is precipitation. Now you know precipitation is a reaction between the soluble antigen with a soluble antibody. So both antigen and antibody, they should be seen into the soluble form. This results into formation of antigen antibody complex, which is called as lattices. And this lattices are responsible for precipitation, or the reaction is called as precipitation reaction. So here it is represented diagrammatically. All the square check boxes, these are defining or they are showing the antigens, whereas these are the antibodies. So both antigens as well as antibodies, they are seen into soluble conditions. So these soluble antigens and antibody, when they are showing similarity between their epitope and paratope, they get associated with one another. The reaction is reversible, and this leads to formation of a complex which is much more larger in size as compared to individual antigen. And this leads to bring about formation of a large molecule, and this, because it is much more larger in size, it tends to settle down or it tends to precipitate, and this reaction is called as precipitation reaction. Now this precipitation reaction can be quantitative in nature, and this totally depends on the amount of antigen which is mixed with the constant volume of antisera. Obviously, I can say the precipitate could be measured, and this is totally dependent on the ratio of antigen to antibody. Also, it is dependent on the avidity of the antibody. Of course, what is avidity? Avidity is the total capacity of the antigen to interact with the antibody. That is called as avidity, right? Now, when a plot is made to understand this quantitative precipitation reaction, we can say that there are three different zones, that is zone of antigen axis, then zone of equivalence, and the zone of antibody axis, right? Now what is zone of antigen axis? Now here it is shown, and when the antigens, they are seen in large quantity as compared to the amount of antibody, or in other words you can say here in zone of antigen axis there is less volume of antibody. As a result, what we can say, the complex which will be formed, they will be very less, right? And that is why when you plot a graph, you will get the line or the curve which is minimal in nature. What is zone of equivalence? The middle one is defining the zone of equivalence. As the term equivalence says equity, it means that probably the number of antigen and the number and the amount of antibodies, they are sufficient enough to show interaction, and thus there is observable antigen antibody complex can be formed, and this can lead to bring about precipitation. When the antigens, they are excess and antibodies, they are less in volume, obviously the precipitation reaction is not visible. In the third case, when antibodies are more and the antigens, they are very less, even in this condition, the interaction between antigen and antibody which occurs is not seen or it is not visible because of variation in the ratio of antigen to antibody. Thus, here you cannot see precipitation. So, from this graph, what you can recall or you can remember that both in case of zones, when antibody is in excess or antigen is in excess, in this condition there is no precipitation reaction observed. It is only the zone of equivalence which will show maximum amount of precipitation reaction, because the antigen antibody ratio is seen into equilibrium, right? This is once again shown into the graphical representation, right? Here you can say antibody is seen in excess, antigens they are less. So when it is very less, the binding it will go on in linear progression. At zone of equivalence, there is no free antigen and free antibody, because in this zone of equivalence, both antigens and antibody they have binded together, resulted into formation of immune complex or antigen antibody complex. And once again, when antigen is in excess and antibodies they are in less quantity, there is obviously decline in the antigen antibody interaction, or you can say the complex they are less seen, hence the rate of precipitation will be lowered. Now let us talk about agglutination. Agglutination is seen when antibodies binds and cross-links the cells or particles. As a result, a clump is formed, and this clump is always seen in aggregated condition. Or in other words, you can say that here the microbial organisms which are invaders, they are seen entrapped, and this entrapment is seen commonly with two different types of immunoglobulins, that is IgM and IgA. IgG is sufficient to bring about the process of agglutination. Now here you can see all these hexagonal structures, they are representing the virus, whereas these Y-shaped structures they are the antibodies, right? So antibodies they interact with the the paratope of antibodies, they interact with the epitope of the antigen, and as a result there is formation of a clump, or there is formation of an aggregation, and this aggregation is called as the base for agglutination reaction. This you must have performed in blood typing. You could see the presence of clumping of blood or agglutination of blood when antigen and your blood and antisera they are added together, and that defines the blood type of the individual, right? Here once again I have tried to define the epitopes which are seen present onto the surface of bacterium, that is nothing but an antigen, and all these green shaped structures, these are the antibody. This is a pentameric antibody, that is IgM, and its specificity enables it to choose the specific type of epitope, and that causes the clumping, or that results into the process which is called as agglutination. Now this is once again the same structure as I mentioned, that when you are doing the blood typing, these RBCs they show surface receptors which are seen interlinking with the antibodies, and that results into formation of a clump. So this is the antibody interacting with specific type of the receptor molecule onto the surface of RBC, and that enables us to define the blood type of an individual, right? Now what is the applications of these agglutination? Agglutination is used in quantification of antibodies, and it is also used in determination of blood type. So the blood type is this reaction in which the blood typing is done is called as heme agglutination. The second is quantitative agglutination. test, and this helps in measuring the levels of antibodies against a particular antigen. That means the titer of antigen can be found out with the help of agglutination reaction. There is passive agglutination reaction wherein erythrocytes are coated with a soluble antigen. Now this soluble antigen could be a viral antigen, or it could be a polysaccharide, or it could be an hapten, and is used to coat the RBC. And this coated RBC brings about the agglutination test for an antibody to the soluble antigen. So this is called as passive agglutination. So this is the pictorial representation or diagrammatic representation of passive agglutination. Y-shaped structure is soluble antibody. Now these red spheres, they are defining the RBCs or erythrocytes, and the blue small structures what you can see, these are the antigens. These antigens are forcibly seen, loaded or coated onto the surface of or to the surface of RBC, and as a result of which these antigen antibody interaction which is seen formed enables us to understand the amount of antibodies which are seen present into the serum, right? And third one is Coombs test, that is antiglobulin test. Then comes the third one, neutralization. Neutralization is actually the binding of antibody to microbial epitopes. Now what is epitope? Again epitope, and it is seen written with microbial. So this way also you can remember that I am talking about the antigen. So binding of antigen to antibody which are both seen in soluble conditions and which inhibit the binding to the host cell is cause of the neutralization reaction. Or you can say that whenever the epitopes of antigen binds with the soluble antibody, that will bring about the nullifying effect of antigen, and therefore it is neutralization reaction. This is usually shown by the antibodies like IgG and IgA, and it is used in identification of certain diseases. Opsonization is the fourth type of antigen antibody reaction in which a pathogen is marked. So when we say pathogen is marked means I am going to tag the pathogen. What is tagging? Tagging is made to visualize the presence of certain particles into the vicinity. So we are tagging the pathogen which is ingested, and now this tagged pathogen, when it is destroyed by the phagocytic cell, it can be visualized very easily. Now this is what I meant to say with reference to opsonization. Now suppose this is the bacterium. Now this bacterium will be identified by the soluble immunoglobulin, that is IgG. Obviously what I can say that this IgG, when it binds with bacterium, it is getting tagged, right? And now this tagged bacterium will bring about activation of the complement, and this activated complement will result into development of certain receptors onto the surface of antigen. That means here you can see, compare the structure of bacterium initially before tagging and after tagging. So once it has been tagged with IgG, it will activate certain development of proteinaceous component which are seen expressed onto the surface of antigen, and these expressed proteins will be identified by the phagocytic cell, and that leads to, or that will initiate the process of phagocytosis, and to have this phagocytic action on larger extent, it is very much essential that the antigens, they should be tagged, and this tagging is bringing together all the pathogens in the close proximity of the phagocytic cell, which leads to phagocytic action. Now, the fourth one is antibody dependent cell mediated toxicity, which is called as ADCC. A stands for antibody, D stands for dependent, C stands for cell mediated, and the second C stands for cytotoxicity, that is antigen dependent cell mediated cytotoxicity, or ADCC. So here the coating of organism can attract the phagocytic cell as well as other cytolytic cells like the natural killer cells, eosinophils, macrophages, and so on. Now, this organism which I am talking about and is coated, it could be any bacterium, protozoan, parasitic worm, or sometimes even the cancerous cells. So such cells or such antigens, these are used for the cytolytic action, and as a result of which the deformed cells or the antigens, they are killed. And when such action is observed, it is called as antibody dependent cell mediated cytotoxicity. So why is it called as antibody dependent? Because as I said, the organism should be coated. So with what the organism will be coated? It will be coated with the antibody. Look at this diagram. You just see this oval green structure. This is an antigenic representation. So this antigen will get associated with IgG, that is the antibody. So obviously now antigen is being tagged with, it is labeled with large number of antibodies. Now when this is the condition, then this cell, that is the antigen, it will be easily identified by the other bodily cells that are phagocytic cells, like the mast cells, you can say, the natural killer cells, or the eosinophils, or dendritic cells, and so on. So as a result of which, these phagocytic cells, they get in association with the labeled antigen, and these labeled in the sense coated antigens with the antibody, and this causes the cross-linking, and the cross-linking results into degranulation of the mast cell, and because of degranulation, whatever the cells are seen coated with the antibodies, they are you can say fragmented, or these cells, they show the phenomena of mortality, or they are brought to apoptosis action. Now, destruction of large parasite. Now, as in the previous slide, I was talking about the smaller pathogens or the soluble pathogens. But what about the parasites which are large in size? So, a large parasite will also have large number of receptor molecules onto their surface, which will be identified by the IgG molecule. Now, all these green Y-shaped structures, these are IgG molecules. They will be identified by the receptors. After identification, there is a cross-linking seen between the antibody and that of the receptors of antigen, and this will activate the eosinophil or the natural killer cell. Here the activation, here the action is shown with reference to eosinophil. So attraction of eosinophil will enable the eosinophil to give out large amount of perforin type of component or the secretions, and these perforins are actually the proteinaceous component which are containing large amount of lytic substances within itself. So these perforins, they will be seen secreted in close vicinity with the larger molecule, that is the parasite. And here you can see there is defragmentation seen onto the surface, because the perforin, it will bring about formation of pores onto the surface of this antigen. As a result, the natural component of the parasite, it will be seen oozed out, and that will bring about the death into the parasite. So this is one way you can say. The second is the macrophages also. When you talk about the macrophages, the action is similar to that of the eosin, but here you have to remember that large amount of cytokines will be secreted. Now these cytokines, they contain the lytic enzymes, and these lytic enzymes, they have the similar effect as that shown by the perforin released by the eosinophils. Now with this, I conclude the session, and in the next class, I'll be talking about the different tools like immunoelectrophoresis, immunofluorescence, and ELISA. Till then, stay safe, stay healthy. Thank you.